Hi everyone, welcome back to my channel. In the previous video, I discussed regarding transcription in the case of prokaryote. In continuation with that, in this video, I'm going to discuss about transcription in the case of eukaryote. As you all know, transcription is a process of copying of genetic information from one strand of DNA into RNA, or in simple term, it's a synthesis of RNA from DNA. The process of transcription in the case of eukaryote can be studied under mainly three headings, that is initiation, elongation and termination. We'll discuss these three steps one by one. First step is initiation. The synthesis of RNA or the process of transcription begins with the initiation step with the help of enzyme known as RNA polymerase. RNA polymerase is also called as DNA dependent RNA polymerase. Unlike prokaryotes, in the case of eukaryote, there are three types of RNA polymerase. They are RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. These three types of RNA polymerase has the ability to produce different types of RNA. As you can see, RNA polymerase 1 will produce rRNA, RNA polymerase 2 will produce hnRNA or precursor mRNA, RNA polymerase 3 has the ability to produce tRNA, 5S RNA and snRNA. All these type of RNAs are essential during the translation or protein synthesis process. So because of which these proteins will be produced during transcription process with the help of this RNA polymerase enzyme. And that synthesis of RNA begins with the first step known as initiation. The initiation step begins with the binding of RNA polymerase to the promoter region of the transcription unit. In the case of eukaryote, there are several special type of protein known as transcription factor. It is this transcription factor which recognizes the promoter region of the transcription unit. Each RNA polymerase have their own transcription factor. For example, TF2A, TF2H, TF2D, etc. And it is this transcription factor which recognizes the promoter region. Once the transcription factor recognizes the promoter region, the RNA polymerase enzyme go and bind to the promoter region along with the transcription factor. The complex that is formed as a result of binding of RNA polymerase to the promoter region along with the transcription factor is known as pre-initiation complex. As soon as the RNA polymerase go and bind to the promoter region along with the transcription factor, it catalyzes the unwinding of the DNA double helix which provide free DNA template strand for the addition of ribonucleotide. That initiate the second step, that is elongation. As soon as the initiation, the pre-initiation complex or some of the transcription factors are released from the RNA polymerase which initiate the second step, that is elongation. During elongation step, the RNA polymerase will move along the DNA template strand and for each base of the DNA template, the ribonucleotides are added by RNA polymerase based on the complementarity. This addition of ribonucleotide by RNA polymerase will elongate the RNA chain. Apart from addition of ribonucleotide, it also adds special type of nucleotide called as 7-methylguanosine. So during elongation step, 7-methylguanosine is begin to add to the 5' end of the pre-mRNA. This addition of 7-methylguanosine or capping will protect this pre-mRNA or growing mRNA from the enzyme. Apart from that, during elongation step, in the case of eukaryote, it involves special type of factor called as elongation factor. This elongation factor will enhance the overall activity of the RNA polymerase. So that is all about the elongation step. Next step is termination. The process of termination in the case of eukaryote is different for three different types of RNA polymerase. For example, in the case of RNA polymerase 1, termination of transcription occurs when RNA polymerase 1 enzyme encounter a binding site for special type of protein called as Reb1p protein. So when RNA polymerase 1 enzyme encounter this binding site for Reb1p protein, it causes termination of transcription. Second one is RNA polymerase 2. In this case, the termination of transcription occurs just by cleavage. So 3' end of the mRNA that is produced is by endonucleolytic cleavage of the mRNA. In the case of RNA polymerase 3, termination of the transcription occurs when the RNA polymerase 3 enzyme encounter a sequence, that is stretch of time in sequence followed by self-complementary sequence on the DNA strand. When RNA polymerase 3 enzyme encounter this sequence, it causes a termination of transcription. So that is how termination of transcription occur in three different types of RNA polymerase. At the end of termination of transcription in the case of eukaryote, three types of RNA are produced, that is mRNA, gRNA and rRNA. These three types of RNA that is produced are not mature RNA. They are called as primary transcript or hnRNA. These primary transcript or hnRNA now undergo post-transcriptional changes or RNA processing to become a mature RNA or mRNA. Post-transcriptional changes or RNA processing takes place mainly by three steps, that is capping, tailing and splicing. We'll discuss these three steps one by one. As you can see in this diagram, this is a hnRNA or primary transcript that is produced. During the first step, that is during capping step, a cap or a special type of nucleotide called as 7-methylguanosine triphosphate is added to 5' end of the hnRNA or primary transcript. Addition of this cap will protect the hnRNA or primary transcript from the nucleases and the other enzyme. Apart from that, it also helps during the initiation of translation or protein synthesis. Anyhow, the capping process has begun during the elongation step itself. That is regarding the capping step. Next is tailing or polyadenylation. As the name indicates, in polyadenylation of about 200 to 300 adenylate residue is added to 3' end of the hnRNA or primary transcript. The tailing process will stabilize the hnRNA or primary transcript. So that is about the tailing step. Next is splicing. As you can see in this diagram, the hnRNA or primary transcript consisting of both introns and exons. Introns are the non-coding sequence. Exons are the coding sequence. During transcription, both introns and exons are transcribed as RNA. During splicing, these introns, that is non-coding sequence, is removed and exons are joined to form a functional mRNA. And it is done by a splicing apparatus called as spliceosome. This spliceosome consisting of several small nuclear ribonucleoproteins, which is abbreviated as a SNRPs. So this spliceosome will remove introns and it will join the exons to form a functional mRNA. This functional mRNA, which is formed as a result of splicing, is much shorter when compared to the primary transcript or hnRNA. Now this functional mRNA will be transported out of nucleus for the next process, that is translation. I hope this video will be useful. If you like this video, please hit the like button and subscribe to my channel. Thank you.